Okay, lecture 13, looking at lipids. Talking about structure, it also talks about other issues such as rancidization, which more fairly soon. Yeah, just to remind ourselves first of the classification of them, and look at rancidity for quite a while, then finish up by talking a bit about the effect on structure. Okay, so classification, but first, here's a question to start with. There's a list of calorific values. Why does fat have the highest calorific value? Pause the video and you have a think about this. As you may recall, we may have done this in human metabolism. Energy metabolism is based on 2 carbon fragments, which all of these produce. So why is the energy value for fat higher than the others? Right, so I'll assume we've had a little think about that. It's because lipids are very hydrophobic, and in fact they're very largely anhydrous. This can be seen clearly in adipose cells that are essentially fat reservoirs surrounded by the rest of the cell. In contrast, both carbohydrates and proteins extensively hydrogen bond with water, so reducing the overall proportion of molecular weight involved in energy metabolism. Similarly for ethanol, which is in effect infinitely soluble in water. Okay, so here's a general classification. Although the other classifications have important biological roles in food terms, we're mostly interested in fats and oils. Wax is rather less. Saponifiable, what does that mean? Saponification is the hydrolysis of an ester with a base, which often makes a soap-type product, or a soap. And there's the soap. Okay, moving on. Distinguishing between oils and fats. Oils have relatively short chain fatty acids, whereas fats have relatively long chains. Generally speaking, when you increase molecular weight, this increases melting point, which is what happens here. This is why plants produce oils, because they are lower melting point, like the environment which they live in, and animals that metabolize at a higher temperature produce fats that are solid at room temperature. It remains an open question whether this is a real advert. Okay, just to simplify matters, when we draw long hydrocarbon chains like these, it's convenient to exclude the hydrogens, which are there still, of course. So the last couple of carbon atoms have a CH3 and a CH2 group, but for simplicity, we just don't draw them in. So when you see these zigzaggy lines, they're referring to chains of hydrocarbons. Okay, if we compare the structure of fatty acids with those of fat-soluble nutrients like beta-carotene, which I'll look at a bit later on probably, both have long chains of carbon atoms. There's a principle that says like dissolves like, which is why things like beta-carotene will dissolve in fats. But here's some more definitions. So if you look at a fatty acid, a long-chain carboxylic acid, it's got the aliphatic chain, which is hydrophobic, so it doesn't dissolve in water. And the carboxylic acid head, which is hydrophilic and will dissolve in water. So they're they have 2 characteristics really, which is quite important in their structure. Okay, we'll look at triglycerides, simple lipids. The base development is glycerol backbone with 3 long-chain fatty acids that say carboxylic acid groups bound to it. So glycerol is a colourless, odourless, viscous liquid, sometimes called glycerine. And as I said, it provides the backbone for triglycerides, to which are attached 3 fatty acid chains. The ester group is often drawn in the simplified form shown in the main diagram, but don't forget, however, the structure is a little bit more complicated. When drawing chemical compounds, always make sure you take account of how many bonds elements actually form. So for carbon down here, it forms 4 bonds. So the esterification reaction. Esters are formed by a condensation reaction between an alcohol and a carboxylic acid. Note the loss of water. This is typical of biological reactions that join 2 compounds together. So the synthesis of amino acids, the synthesis of proteins from amino acids, for example, involves the loss of water. And conversely, when you break them up, you hydrolyze them, the structure gains water, effectively. Okay, so another definition, saturation. No double bonds in the fatty acid chain. Note this definition does not consider the oxygens in the ester group over here. Point is gone, there it is. just whether there are any in the carbon chain. Saturated fats tend to be high melting point compared to unsaturated ones and often obtained from animal sources, as mentioned early on. Consumption is linked to a higher incidence of cardiovascular disease. Unsaturated fats have at least one or more double bonds in their carbon chains. Typically obtained from plants and liquid at room temperature. They are not linked to increased levels of cardiovascular disease. Note the kink in the chain produced by the double bond. Okay, so naming these things in nomenclature, if you're really interested, you look at the International Union of Pure and Applied Chemistry Information or any food chemistry textbook. Generally we use simple, trivial names such as linoleic rather than the full systematic names, and I think it's easy to see why here. One thing that is important is notice where the carbon chain starts. In this case, the carbon chain starts with the carbon and the carboxylic acid group. Sometimes though we use the omega nomenclature. In this case, the numbering starts at the terminal carbon atom of the chain. So omega is 1, and alpha over here is whatever it happens to be. Again, ends at the carbon atom. I beg your pardon. Yeah, no, that's right. Yeah, that is right. Yeah, ignore me. Okay, so going back to the packing, going back to the consequences of the kink in the chain and not having a kink in the chain. Saturated fats pack closely together and tend to form solids, whereas unsaturated fats pack loosely and tend to form liquids. This is why saturated fats melt at higher temperature than unsaturated ones. They can pack more closely together compared to unsaturated fats. Okay, so here's a question. A reindeer which has both saturated and unsaturated fats present in his legs. Now, where would you expect to find the balance of them? So as ever, pause the video and have a think about this and see if we can enlighten our curious looking reindeer. Okay, right. Increasing, well, the key factor is here, increasing unsaturation, that's to say double bonds, keeps lipids fluid close to the frozen ground. So the fatty acid in reindeer's legs becomes more unsaturated closer to the hoof, which helps prevent the membranes from freezing. Reindeer legs do have more unsaturated fatty acids closer to the hooves, which keeps the membranes nice and loose. That's according to the University of Alaska's Institute of Arctic Biology. Too bad Santa doesn't have more unsaturated fats, I wrote here at some point. A striking exception in this respect are leg bone marrow lipids, which contain a high proportion of unsaturated fatty acids in reindeer and other ruminants, and seem to be preserved until the last phase of undernutrition, so associated with not a lot of food being available, I guess. There are some links in the notes if you're interested in this. Okay, this is just summarising what we've just discussed before. Right, so up to 8 minutes. I think we'll pause here and come back with a separate video on rancidity.